Hello everyone and welcome to the latest IFST webinar entitled Using Genetics to Understand the Factors Influencing Food Liking and Their Role in Food Choices, which is organised by IFST's Sensory Science Special Interest Group. My name is Stephanie Mitchell. I'm Senior Sensory Manager at PPL Insights, an independent market research agency which is based down near Gatwick. I'm current chair of the IFST's Sensory Science Special Interest Group. Now, this group was founded over 20 years ago, and it allows members interested in sensory science to engage and share knowledge with fellow professionals. We cover a wide range of topics relating to the sensory properties of food and drink, including sensory testing and analysis, consumer trends, neuroscience, the impact of social networking, promotion of ethical and professional standards, and health and wellness. The SSG are also involved in accreditation and examination of sensory training with both foundation and intermediate courses. We've also set up a register for sensory scientists, and this is the only one of this kind in the world. In addition, we have a working group for students and early career researchers. So back to today. This webinar is being recorded and it will be uploaded to IFST's Webinar Hub. Please feel free to make comments and ask questions throughout the webinar using the Q&A function, which should be at the bottom of your screen. This can also be used in the case of any technical difficulties. You can also upvote other people's questions by clicking the like button next to the question. There will be a Q&A session after the presentation where we will respond to as many of those questions as possible. And Dr. Victoria Whelan is one of our committee members. She will be asking the speaker your questions. Our speaker today is Dr. Nicola Piratstou. Since he graduated in biology, his research has been focused on understanding the genetic basis of complex traits. Although his work spans many different areas, his main focus has been on using genetics to understand the factors underlying food choices and to understand the effects of diet on health. Nicola will present the results of his research conducted on genetics and liking for a wide variety of food and drinks, and this knowledge will provide insight into the relationships between food likings and their underlying biology. For more information on Nicola's biography, please have a look at the website, the IFST website. And so with that, I will hand over to you, Nicola.
Hello, I'm Nicola Piratstou, and I'm a human geneticist. I am at the moment working at the University of Edinburgh for the next 10 days about, and I'll be moving in Italy very soon. So today I'm just going to talk a bit about my latest research in genetics and its role in determining food preferences and what role it has then on human health. Just a bit of background. So this is a plot which I've taken from a study which is called the Global Burden of Disease. And basically, these are all the common risk factors which affect human health. Here you have the difference in prevalence between 1990 and 2015. And here you have how important these risk factors are. Now, as you can see, we've been able to reduce the prevalence of the important ones, like smoking or underweight and so on. But one thing we've been very bad at is obesity, which instead of decreasing, has actually increased in the last years. Now, when we think of obesity and biology of obesity, it's a very complicated process which spans from food production to physical activity, the environment, your physiology. And if we think of genetics, it's been really focused on physiology. so hormones, for example. But from recent studies, we know that genetics involves, you know, a much broader number of factors than we thought before. For example, you know, how much you exercise, or, you know, your psychology, your underlying you know, psychological state. But, you know, my niche is understanding the role in determining food choices. Why do I focus on food choices? For two reasons. One is to understand, of course, the biological basis, which we know very little of, but also because, and I'll show some examples later, we can use genetics to establish causal, not correlations, but causal relationships between food and health status. And of course, the final goal is, you know, stratified education intervention based on genetics, maybe new products which are more healthier but also tastier, personalized diets, new medication possibly, but also better, you know, public policies. Now, when we think about nutritional nutritional epidemiology, you know, the big question and always been, what should we be eating? But I think that this question has been answered quite extensively. So while we've been relatively good at understanding what we should be eating, and we keep changing a little bit, you know, a bit more salt, a bit less salt, but more or less we know what healthy food looks like, I think we should be focusing on how to get people to eat an healthy diet. So not what it looks like, but actually how do we get people to eat more, let's say, fruit and vegetables, or, you know, or eat less meat. So how do we improve food choices? Now, in a world where food availability is not an issue, food preferences are definitely, and this has been shown a lot, the first factors driving food choices. Of course, it's not the only one, but if you don't like something, it's really hard that you'll eat it. Now, one question people don't think enough about is, why do we have food preferences? So to try to explain this, I'm gonna use a vampire. Now, the vampire has very limited food choices because all he eats is blood. Right? So vampire doesn't need to eat to have food preferences. It just needs to know if something is blood or not. But in the case of humans, we're omnivores. We can eat anything, right? So our taste and our food preferences have evolved to allow us to choose between the foods we can eat or not. So let's imagine you're a caveman and you're walking, you know, around the forest and you find these two berries. So which one should you be eating? Well, taste certainly guides us. So if it's sweet, it generally contains energies, while if it's bitter, it's usually poisonous. It's not always poisonous, but in a world with no supermarket, it likely will be. So if we think of the basic tastes, they have a specific encoding, right? So sweet will identify sugar, umami, mostly proteins, and then we have bitterness, which protects us, you know, mostly from getting intoxicated, and similarly, the acidic taste usually is associated to spoiled food. The main point is that these two tastes usually indicate energy, and it tells our brains to eat. While these other two is, you know, this is poisonous, do not eat it. Of course, when we think of healthy diet, or, you know, our diet today, this is what sweet and, you know, savory looks like, and this is what bitter and acidic. And basically what we're trying to do is make people eat more what their bodies is telling them not to eat. Okay, this is a really big simplification, but it is just to set the stage. Now, everyone, I get a lot of the fact that, you know, you eat what you're exposed to, right? But if we take, there's been studies on babies, this is done in US, of course, you cannot do this in Europe, where they gave newborn who've never tasted even their mother's milk, sweet solution, they tend to smile, and if you give them bitter solution, they'll make a disgust face, okay? And this is quite conserved across different species. Okay, so if you give even a mouse a bitter solution, okay, then it will react by making a disgust, mouse version of a disgust face, of course. And if you ever try this at home, you can take your baby and give it lemon, and they'll all, or more or less, react the same way. Okay, so there is a very strong encoding. This is not learned. This is, you can try this across the world. This is an encoded biological response, okay? The way this works is that when you eat something good or tasty, your pleasure centers flare up and you get hungry. Well, if you try this with a salad, then this doesn't happen. So you don't get the same pleasure, and certainly you won't get hungry. Of course, this worked very well when this is how we got food, but then today we have a different way of getting our food, and so we have obesity is rising constantly. So when we think about food liking, we should many people focus a lot on taste or flavor, but we know there's lots of things involved, from culture to color to texture. And most of this are the interaction between one's environment, so where we grew up, but also genetic background. Okay, so the genetic background kind of sets the stage, and of course, if the environment is right, then you get a certain reaction. Okay. Now, just a very small side step to explain how we study genetics. Okay. So what I do is called a genome-wide association study, where what we try to do is to understand which parts of the genome are associated to a certain trait. Okay. So let's imagine we have height, and we want to study if a genetic variant is associated to it. We usually use genetic variants which have only 2 alleles, so you can find a variation which is only in 2 forms, either A or B. And then we sort people based on this, and we see if there is a trend. So the more people, the more you have Bs, the taller you will be. And a genome-wide association study, what we do is that we do this between 500,000 and 25 million times. Okay, so this is lots of statistical tests, but they're all very simple. Okay, so they're just simple linear regressions. What you get out of this is what we call a Manhattan plot, which looks like a Manhattan a little bit. And every time, so this threshold is a p-value of 5 × 10⁻⁸, and everything that's over this line we think is significant. Okay, and they correspond in this case to these genes. Okay, so this would be chromosome 1, 2, 3 up to 22, and each of this dot is a genetic variance, and here we have about 11 millions. Okay, now given that the effects we generally study are very small, and also we're doing all of this statistical tests, one thing we need is lots of people to study this. So you cannot do this kind of study with 1,000 people. You cannot use even 10,000 people. Okay, so it's not very easy to come by this kind of data. Luckily, in UK, there is this study, which is called UK Biobank, which has collected 500,000 people for which we have lots of information. Okay, so we know what medications they're taking, we know their GP data, we know if they went to the hospital, we know lots of stuff. But most importantly, we have their genotypes, and we have their emails. So we developed a food liking questionnaire, which was very, very simple to answer to. So it's just how much you like liver, for example, from 1 to 9. This is a simple hedonic scale. And we got back 182,000 responses. This questionnaire is about 150 items. They're all very specific, and they include things like exercising, or we asked about cigarette liking, we asked about watching TV. So all health-related items, okay? But also we asked a lot of food items. So when we looked at the food items and we focused on them, of course, the first thing we did was to see how they cluster together. So this is just the correlation matrix. You cannot read the names, they're too small. But that's just to show that what we found, and we used a thing called the genetic correlation, which basically looks at how much of the genome these traits share. So if they are affected by the same genes, it's fundamentally. As you can see, there's two big groups which form. Now, when we look, but the intergroup correlations, they're not constant, and they are quite weak. They're much weaker than within the groups. When we look at this in a hierarchical clustering way, you can see that we have two main groups. One is this group here, which includes many things which are highly palatable and with high calories. So we have sweet foods, we have meat, we have sweet coffee drinks, kind of Starbucks kind of things. And then we have other things like pizza or crisps, fried stuff, and so on. And then we have another big group, which is a very complex and has lots of stuff, which ranges from healthy foods to things we learn to like, for example, alcohol or spicy foods, bell peppers, all foods which have a strong taste and we have to learn to like. Now, what's interesting is that if we look at these groups, they don't cluster as we would cluster them based on commodity classes, for example. So if we look at vegetables, for example, we have two groups which are very far apart and they don't correlate with each other. Where here we have more like salad and raw kind of vegetables, while here we have other things like aubergine or asparagus, spinach, so more strong tasting vegetables, okay? Now, to try to formalize this, we use a thing which is called genomic structural equation modeling, which is just a way to find dimensions which describe groups, okay? And we applied this in we created a multi-level map, making bigger and bigger groups. And what we found is that we get, this is the final map, we get these three very high dimensional group of foods, where one clusters the foods which have lower calories, so like fruit or vegetables, salad vegetables, and healthy foods. Another one which has all the foods you have acquired tastes, like alcohol, coffee, or vegetables with strong taste. And then we have this group here, which have all high palatable groups, foods like meat or sweets or carbohydrates. Now, the interesting thing is that when we look at the correlation of these dimensions with each other, while the low caloric and learned groups have a relatively strong correlation of 60%, the high palatable groups have no correlation. So there are two big, this suggests that at least from a genetic point of view, these are, and a biological one as a consequence, liking highly palatable foods and liking other foods are two different processes. One which is likely more wired in your brain and innate, and another one which is more the result of acquired taste and learning. Okay. Now, to kind of try to validate our results, which is not straightforward, we compared with consumption of foods for which we had the same result. Okay, so for example, we had also pork consumption or red wine consumption. And overall, we see that the correlation are always very strong, and they're around 75%, which is kind of expected. Meaning that we're actually measuring, well, we think this is a good indication that we're measuring what they like. Now, the interesting thing is that when we look at the heritability, which is how strong the genetic component is, the heritability, these labels are wrong actually, but the heritability of liking are twice as big as the one of consumption. So it means that we are measuring something very similar because the correlation is high, but yet we're closer, we're one step removed toward biology, which is what we're interested in. Another thing we can do is we can profile these people, seeing the correlation with many other traits which are available from the literature. And in agreement with what we see is that the people who like lower caloric foods, so fruit and vegetables, they're also taking more supplements. They're exercising much more, and apparently they're happier, and they have a higher overall rating. The other thing is that they also have lower sodium in urine, which means that they're actually eating, a lower sodium diet. Well, when we look at the people who like highly palatable food more, they like, they're much more sedentary. So they like, they spend lots of time watching TV, but they also have higher sodium and creatinine in urine, which means they're also eating more, meat, for example, but also they have a lower educational attainment. The other thing we looked at to try to validate our measures and our results was correlations with brain imaging and brain morphology data, which is available. We did, again, what we call the innate or the highly palatable foods show a completely different patterns from the one which is the learned foods. But most interesting, it shows a negative correlation with the volume of the striatum. Okay, so we have higher liking for palatable foods, lower volume of the striatum. And this is the striatum. And what we know is that it's a very important area of the brain, which through the dopaminergic receptor and opioid receptor is responsible for responses to pleasure in food as well. And what we know is that generally a lower activation is, it corresponds to addiction in general, but also to uncontrolled eating of reward foods, which is exactly what we found. So we found that a lower volume, and in this case it's a lower activation, is also linked to a higher liking of rewarding foods. Now, looking at more specifically at the genes we found, so this is a 3D, sorry, it's a bit heavy. This is a 3D version of the plots I've shown you before, where here you have all the 150 foods, and each of these peaks is a genetic association. This is kind of a bird's eye view. Now, for those of you who are into genetics a bit, this is FTO, for example, which is the obesity gene. ApoE is very well known for Alzheimer's and also for LDL cholesterol. But we do have many genes which are, like this ones here, for example, which seems to span a large number of foods, but have not been described associated to anything yet. Okay. Now, I'll just show you some examples. First of all, these are the 10 top associations we find. As you can see, we have very, very low p-value. This would be 10 to the minus 71, for example. And here we do find things we expect related to flavor. For example, for garlic, we do find that this is an olfactory receptor. This is a non-synonymous variant for olfactory receptor. Similarly, for onion, we have another olfactory receptor. But also we do find the things we expect. For example, ADH1B is very well known to be associated to alcohol consumption, and we do find it in wine. While FGF21 is known to be the sweet tooth gene. Okay, so this is just saying that what we find makes sense given what we know about these genes. Okay, so one of the first observations, and this is what we did, we looked at which traits were associated to either taste, olfaction, or chemstasis, like, I don't know, like for example, spiciness, receptors, to which food they were associated. Now, as you can see, the flavor differences, genetic flavor differences, are all on this side, while there's none on the highly palatable foods. And the reason for this is likely to be evolutionary. Okay, so if we imagine that one of the things in life that's most important is getting energy to survive, reducing anything that reduces liking for calories is not good from an evolutionary point of view. While if we think of these foods, on the other hand, they're generally not liked as a base. So these are mostly taste receptors. We don't have the umami or sweet receptors. So these are all mostly bitter receptors. So reduction in bitterness, although it may expose you more likely to being poisoned, on the other hand, it will broaden your diet and so increase your chances of survival. Okay, so this is a first observation. Now I'll show you some specific results and I'll show you what we can do with genetics. Okay, the first gene I'd like to talk about, it's called FGF21, which is an hormone, and the A allele is associated to liking more sweet foods, fruits, and I'm not sure why, white wine. Okay, so anything that contains sweet. Okay, so for desserts, but also fruit, even though they don't correlate normally. And liking less anything that's savory. Okay, so not just meat, but also fish or cheese or capsicum, for example, strong flavors and vegetables. Okay. Now, what we can do is see, okay, so after it changes your liking, what happens with consumption? And in fact, we do have a very good correspondence where the A allele is associated to higher liking of carbs in diet, higher consumption in percentage in diet, more alcohol units, but also while it's associated to lower protein in diet and lower fat. Okay. Now, the interesting thing is that this is done without any difference in energy intake. So this is kind of an experiment. You can consider this almost as though it was an experiment where people with the A allele for their whole life have been eating more carbs, less proteins and fat without any difference in energy. Now, if we look at the long-term effect of this, these people have a higher waist-to-hip ratio, higher diastolic and systolic blood pressure, likely due to alcohol, but also they have a lower percentage of body fat and they're slightly shorter. Okay, so changing your diet to a higher carbs, less protein and fat leads to very little difference in long-term health effects. Okay, so certainly not BMI, certainly not obesity. Okay, another example, a similar thing we can do about this other gene, which is called GIPR. There is this allele, again, is associated to higher liking of low-calorie food, all of these, and fruit juices, for example. So it's really about fruit and vegetables, while a lower liking of cheese and mayonnaise, but also like cheesecake or cake, right? So it's like really fatty, sweet, mostly fatty foods. Now, again, if we do the same experiment as before, and we looked at consumption, these people are actually, eating more fresh fruit and less cheese, okay? And if we look at the long-term effect, they have a lower BMI, they have lower risk of coronary artery disease, they have a lower body fat, and so on, okay? So changing your diet from a fatty one to a lower caloric one will make you thinner. Okay, so this is not, how can I say that? This is not so surprising, of course. But the interesting thing is what this gene does. So if we look at GIPR, is the receptor of GIP, which is one of the incretins, and which has a lot of different effects around the body. Now, why is this important? Because GLP-1 agonists are used for type 2 diabetes therapy. And they also have a side effect. So this is a trial on type 2 diabetes patients where they were not given any type of dietary intervention. So people with GLP-1 agonists lost about 3 kilograms in 26 weeks. Now, when you use the dual agonist, GIP, GLP-1, people lose up to 11 kilograms over 26 weeks without dieting, okay? And what we've seen is that the gene, which is the receptor for this hormone here, is actually changing your preferences from liking fatty foods to liking low caloric foods. So it's interesting, or it's suggestive, and we should test this, that this drug is acting through changing the foods you like. So you don't have to go through a difficult diet, you just take a pill and then you start eating better through the fact that your preferences have changed. The last example I wanted to talk about, because it suggests another different mechanism, is the alcohol liking gene, which is called ADH1B. Now, In our data, we see a very strong correlation between ADH1B variants and all different, most different alcoholic beverages. Now, the way this gene works, sorry about the flags, I forgot the animation, is that when you drink ethanol, it goes into your liver and it gets turned into acetaldehyde. It then gets turned again by this other gene, ALDH2, into acetate. Okay. Now, in Europe, a variation of this gene makes this process much faster, and so you get accumulation of acetaldehyde. While in Japan and East Asia in general, a variation of this gene here, ADHL2, makes it slow, and so you still get an accumulation of acetaldehyde. Now, what happens basically is that you feel sick. Okay, so you feel really sick. So people who have the fast allele here or the slow allele here get sick from alcohol and they don't drink it. Now, so this is a very specific mechanism which has to do with ethanol. Now, what happens is that when we looked at our data, we did find that the slow allele is associated to higher alcohol liking, but also sweet liking, which makes, from a biological point of view, it's very puzzling. When we looked at, and this is strong, so this is just as big as, the sweet tooth gene, okay? But when we looked at Japan, what we found is that the allele associated to lower alcohol liking was also associated to sweet liking. So if you think at the way it worked, where these two agreed on alcohol liking, this is quite puzzling. So of course, one hypothesis is that this gene has a direct effect on sweet liking, but more suggestive is to think that ADH1B is changing how much you drink alcoholic beverages, and through the exposure to the alcoholic beverages, you change how much you like sweet. So the difference between Japan and Europe would be because the alcohol is drunk through differences in the beverage and the taste. So the exposure to this taste is changing how much you like sweet, which would suggest that it's possible to change liking to one food through exposure to another, which opens, of course, interesting, I think, perspective in terms of interventions. Right. So if it would be great if we could change liking to vegetables by exposing children to something else. So in conclusion, I think we've generated one of the first, comprehensive map of food liking using, I'm not aware of other studies of this size. So this was, I think, and we highlighted this two biologically separated domains. And of course, this is important for the rest of our research, at least. We have, of course, for the first time identified a large number of genetic loci. There were very few known, and now we have more than 200. So that opens, I think, highways for understanding how our bodies and how genetics, how food liking is encoded biologically. And of course, I've shown these mechanisms are very complex. We do need to work much more to understand. I think the ADH1B example shows how complicated this whole process is. But of course, one of the outcomes is that our results also suggest that we could direct food choices using medications. And in the, for the most cases of obese people, the most obese people, that could really help them improve the quality of their diet. Of course, this is just a first study. We need much more data on different ages. This is people between 40 and 60, so they're relatively old. We need other countries, different ethnicities, and so we can understand better this whole process. I need, of course, to acknowledge all my collaborators, and if there are any questions, I'm happy to respond. So I'll give back the floor.
Thank you, Nicola, for such an interesting and engaging presentation that is very relevant to today and the future. And it's fascinating to see the coming together of sensory, physiology and pharma. So I'm Dr. Victoria Whelan. I'm sensory scientist at British American Tobacco and part of the events and communication groups in the SSG. So I'm now going to take you onto the Q&A session. So just a reminder that you can submit questions using the Q&A function, which should be at the bottom of your screen. You may need to hover over the screen if using a mouse or to touch your screen. So we've had a couple of questions come through already. So I'm going to take the first one, which it looks like it's been liked, so bumped up to the top. So that is, if genetics controls food liking, why do I like different things to my sister?
Okay, well, that's, I mean, right. So you are not, your sister is not your identical twin. So you share only half of your genome with your sister. And of course, genetics explains only half of food liking. So we have a study on identical and non-identical twins. And what we know is that the proportion of variance explained by genetics, it's only half. So of course there is a learning experience, but the thing is that the other half is not your family or environment, is what happens, it's your own experiences, is what happens to you. So for example, if you eat sushi and you get sick and your sister doesn't, you won't like sushi anymore and your sister still will. So there is this, it's about the complexity in interaction between what happens to you and your genetic background. And of course, your sister shares only half.
Mm-hmm. Thank you, Nicola. Yeah. Leading on from that question, I just have a question to you about twins. So you said that you've done some studies. Have you done any studies with identical twins that have been brought up in different family situations or different locations, and what effect? Not
yet. You mean like adoptees, for example? They're not easy to come by.
No.
No, I mean, because usually identical twins, they're kept together. So there are examples. One of the first studies, actually, the guy had to make up the twins to publish it because it's really hard. I know there are some in UK Biobank, but we haven't looked yet. But certainly it would be interesting, yeah.
Right, definitely. So another question then that we have got within the Q&A box is, was any cross-referencing looked at with disposable income? So I understand that was in relation to, I think, the chart that you showed.
Yeah. We don't, so no one else looked at that specifically. We do have like Townsend deprivation index. We have other indexes of socioeconomic status, but no. So that's the quick answer is no, but the data is certainly there to be used. So UK Biobank has this thing, it's really cheap to access. It's open to anyone who has an institution behind, so it's worth looking at.
Right. And a question that I had in relation to, I believe, the same chart was, it said for the low caloric people, that they had lower intelligence, or that was my interpretation. So I just wondered what that meant.
Yeah, so I think it's hard to just extrapolate. It's just genetic correlation. I think these are, I mean, people really like low caloric food and nothing else, basically. They're really into it, like they're really doing lots of activity, they're taking supplements. So I think there is kind of a religious kind of background almost. I mean, it stops being completely rational, it becomes kind of a belief system. So I think that's what it's picking up. But the correlation is very small, it's only 15%. So so it's not a strong correlation, but I think it's picking up those people who just are into, you know, really into diet beyond any type of reason. In fact, the interesting thing is that it's not associated in any way to socioeconomic status. So it's really a band across, which spans from, you know, high educated people to lower educated people. They're kind of their own category.
Yeah, which is interesting in itself. So another question then that we've had come through is, could what looks like genetic causation in some cases be back correlation of cultural practices in groups of people from similar backgrounds, for example, similar genetic makeup?
Yeah, well, yes, so we are very careful about that. For example, in the case of ADH1B, we know exactly the physiological, you know, what happens in the liver. It's, you know, we have a very strong biological piece. I do have a paper where I do disentangle. I have a completely statistical method to kind of disentangle the reverse causation, because we have a big problem with food. People don't forget about this, but whatever, people in diet are actually people who have been obese. So if you study the genetics of diet, you're also studying the genetic of being obese. So there is a lot of that. It's possible to take into account. And of course, if we think of the other aspect is that, of course, all these genes are interacting in an environment, right? So if we were to try to study, for example, one thing ADH1B does, it's also increases your blood pressure. So it gives you hypertension through alcohol consumption. Now, if you go to, I don't know, Pakistan, for example, where they don't drink, ADH1B has no effect on blood pressure. So there is a bit of, there is always, our genes are always studied in an environment. So that's why we need diverse populations.
Yeah. Brilliant, thank you. Yeah. So the next one that we've got on the list then is, thank you for a fascinating presentation, Nicola. You mentioned you're moving to Italy. What are your future plans in research? Are you continuing any research with food liking?
Well, yeah, I mean, it took me 10 years to get the data. So certainly I will keep working on this. I intend to work on this. It will be done a bit through collaborations more than hands-on, a bit for a while, but I will be establishing, we plan to establish new groups of people collected in Italy, for example. So we will aim at also, I mean, I aim to keep going on this. I've just started, but certainly I'm open. So I've always been very open to collaboration. So if anyone wants to look at this, I'm very happy to share results, discuss, or, you know, collaborate in any ways. I'm not jealous. I'm not interested in glory. So I'm always happy to work with others.
Well, that's great to hear that you're still going to be working in this field.
Oh yeah.
Albeit in a different location.
Yeah.
So the next question then, we've got, can you see if there is a link between release of chemicals like cortisol and food liking?
Well, yes, we can. We haven't yet. So one thing we can do is find the genes which raise cortisol in people. And because some people have naturally higher cortisol. So we can see if those people have different food likings than the people who have naturally low cortisol. That's actually a very good idea. That's, let's say, on the to-do list. But yeah, we can do that. We haven't done it yet.
Yeah. Okay. So the next question then is, please can you expand on GLP-1 inhibitors? I didn't know it's related to genetics and food choices, and that's something which this person finds fascinating.
Yeah. So specifically of GLP-1, I don't know. GIP. What we found is that GIPR, which is the receptor of the other incretin, which is GIP, is associated to differences in liking. So the way I see the results of the trial is that it may be working through changes in liking more than appetite. This is my extrapolation, of course, it's my interpretation, because no one has looked into it. And before this study, no one had really looked into genetics of liking. There are some studies on TAS2R38. Some of them are, TAS2R38 does not change liking for broccoli. I have to break this to the whole community. But there were very few studies on candidate genes. So this is the first time we looked at all of it. And some of these genes we didn't know. I mean, FGF21 is probably the only case where we knew, but for most of them, we had no clue they were changing liking. So it's certainly something to look into more, I think.
Yeah, that's fascinating. So our next question then is, have you come up with or implemented any interventions for obesity from your gene research of food liking?
Yeah, so not yet. And I'm not sure we will ever be able to use to do diet, based on genetics. I think what genetics can do at the moment, maybe one day we will, but I don't know. But one thing it's telling us, it's giving us, a big insight into biology. And once we know biology, we can start thinking on how to work it. The other thing we may do one day is to separate people who have a problem with, quantity of food from those who have a problem with quality of food, and then design interventions based on that. For example, imagine someone doesn't like, there is a gene which changes liking for coriander, which is pointless itself. But we know what compound is in coriander, which is not good for people who don't like coriander. So all we have to do is make coriander without that compound to make it pleasurable, right? So we can think of this for other foods as well, right? So I don't like vegetables because I find them super bitter. Point is, if I could cook them so they're not bitter anymore, then I would eat them. Right? So, but this can come only through, because most of these things are not communicable. We had a study a while ago where we showed that we use genetics to show that people perceive caffeine more bitter, like coffee more, which means that, first of all, we can tell the bitterness of caffeine from the other bitternesses. But also it means that you can learn to associate a specific bitterness to, the effects of caffeine, and then like it more, even if it's bitter. Okay, but you cannot design an experiment when you ask people how much which bitternesses in coffee you're liking more, because they're all together and we don't even have the words. So I think one day maybe, but we're sorting out things to then go to think of interventions.
Yeah. As a last question, I'm just going to ask you, how do you see the future of genetics and sensory and how they come together?
Yeah, I mean, I think, so first of all, I think what we find is that taste is important, but not as important. It's more about how your brain reacts or how your body reacts. For example, if we take FGF21, right? So it goes up and down based on what you eat. So you eat savory, it goes up. That makes you crave sweet. You eat sweet, it goes down. There's this thing. So I think one thing we should be doing is taking these results to the lab, taking a smaller group of people and seeing why these people don't like certain foods based on that genotype. Right? So is it a certain characteristic? Is it, they're feeling something more? Is it a general thing? And in that case, we can tweak, I think, products to be more liked, healthy products. Everyone who's tried to go into really healthy problem, if you try to make a very healthy cookie, it's hard to make it also very pleasurable. But I think if we were to understand why it's not pleasurable, specifically, not just by trial and error, we may be able to improve it. So I think, so from a sensory point of view, I think there is, I think knowledge to design better products is certainly a way.
Oh, brilliant. Well, thank you so much, Nicola. We have got another question. So I'm going to put it to you and hopefully we can get that one done in a minute or so before we wrap up. So the last question is, have you been able to identify which genes are more prevalent in the different places where the survey's research has been taken?
Oh, no, it's being done in UK. I really haven't looked about, if they stratify. The problem is that if they stratify that way, then it's a problem for the genetic analysis. So we do tend to correct that instead of, trying to exploit it. I do have maps of food liking across UK though. According to birthplace, for example, and there are some differences based on different foods.
Brilliant. Well, thank you so much, Nicola, and thank you to everyone from across the globe for attending the webinar today. If we didn't manage to answer any questions, or if you think of some questions after this session, then you are able to contact Nicola directly. His details are now up on the screen. So we will be sharing a recording of the webinar, including the Q&A session, to everyone that has attended today via email, and our online webinar hub at www.ifst.org/webinars. And I just wanted to again say thank you so much to Nicola and Stephanie and everyone for attending today. And I hope to see some of you soon. Thank you.
Thank you.